Hello everyone, and welcome to McMaster University's Demystifying Medicine YouTube channel. Today we will delve into a recent finding in health research regarding the molecular neurobiology of psychological loss. In this video, we are reviewing a recent study in health research. We will first give an introduction to the study topic, and then provide the background of the research. Following this, we will review the study methods, summarize the results, and highlight the main points of their discussion. At the end, we will wrap it up with the conclusion. For the content of this video, we will focus on a recent study by Smail and colleagues from the University of Cincinnati. This study was published on August 29, 2023 in the Molecular Psychiatry Journal. Smail and colleagues aim to understand how psychological loss affects our brains and behaviors. Now, you may be wondering, what is psychological loss? Well, it is the result of things like grief, whether it be from losing a job, the end of a relationship, the death of a loved one, or other forms of loss. As a result, people often experience a depressed mood, which can resemble a well-known disorder known as major depressive disorder, or MDD. This can lead to a lack of motivation, sadness, rumination, withdrawal, and weight gain. It can be quite challenging for researchers to study the biological aspects of loss in clinical settings, so these researchers chose to study the phenomenon on rat models. They housed rats in an enriched environment where they would feel happy, but later, took the enrichment away to stimulate these feelings of loss. The researchers examined an area of the brain known as the basolateral amygdala, which plays a major role in stress and loss. This is an area of your brain that helps you to feel and remember emotions, particularly fear, and it helps you adapt to stress. So if you're being chased by a bear or walking into a job interview, you can thank your amygdala for giving you those nervous feelings. They also took a molecular approach and found changes in the brain's microglia and extracellular matrix. Now, what are those two things that I just mentioned? The microglia are the protector brain cells. They are on constant patrol for pathogens, or invaders, as well as damage to the brain. So, they're the bouncers of the brain. The extracellular matrix is essentially the architectural framework, or glue, that holds the brain together. It is pivotal for brain function and development, supporting cell-to-cell connections, and repairing brain tissue after injuries. When Smail and colleagues removed this extracellular matrix, or glue, in the rat's brain, they found that the rats started to behave more normally, even after experiencing loss. So let's take a look at the researchers' methods for this experiment. They used 8-week-old male rats and randomly assigned them to one of 3 groups: one with regular housing, one with an enriched environment, and one with a removal of an enriched environment. They conducted 6 different experiments and measured the brain's responses to these changes. They focused on and collected samples from the basolateral amygdala. To further investigate the molecular changes in the brain, they created a new group of rats where they could investigate their genes and proteins through numerous memory and reaction tests. Additionally, they conducted treatment in one group of rats to see if they could prevent loss-associated behaviors, and compared it to another group that didn't receive this treatment. To collect and analyze all this information, they conducted tests such as immunohistochemistry to look at the proteins in the samples. With this, they can essentially color proteins so that they can be seen under a microscope, and the researchers can understand what is happening in the tissues. Well, let's take a look at each of the 6 experiments. In the first experiment, they found that when the rats were removed from the enriched environment, it showed changes to the basolateral amygdala, the fear center. In the enrichment removal group, or loss group, they found an increase in cells and activity compared to the enrichment group. This is due to the associated stressors and emotions that are triggered following loss. In the second experiment, they decided to look at the genes in the basolateral amygdala. They looked at the 3 main groups of rats that I previously mentioned: the normal, enriched, and enriched to deprived environment. They discovered that in the group where the enrichment was removed, there were genetic changes to the cells involved with the immune system and communication between cells. The microglia, or bouncers, were less active, and the extracellular matrix, or glue, was increased. Within the extracellular matrix, there is something called perineuronal nets, or PNNs. These are the safety nets of the brain that wrap around the cells to protect them. So, following this loss in rats, there was an increase in the safety nets hugging the cells. In the third experiment, the researchers used different methods to collect data, but found the same results as the second experiment. In In the fourth experiment, they used immunohistochemistry, or protein coloring, to confirm these changes. They were able to find that the microglia, or bouncers, became less active, smaller, and less complex, but there was an increase in the safety nets, or PNNs. In their fifth experiment, the researchers performed fear-related tasks on the rats. The group that had the removal of the enriched environment, or loss, had issues with remembering and adapting to fear, which is in line with the previously noted changes to the basolateral amygdala. They would spend more time in the threatening environment in comparison to the other groups. By the sixth experiment, they wanted to see how much impact these safety nets had on the rat's behavior. So they brought in an enzyme that broke down these PNNs. What do you think happened? Well, they found that after breaking these down, the rat showed fewer behavioral effects from loss, further emphasizing the impacts of PNNs. So, we just threw a whole bunch of information at you, and you must be wondering, what does this mean? Well, a major finding was the role of the basolateral amygdala in loss. The microglia, or bouncers, became less active. They stopped paying attention to and interacting with all of the intruders. The extracellular matrix, or glue, and particularly the safety nets, or PNNs, increased. So what does this mean? Well, the brain would have decreased plasticity, meaning it would be unable to properly adapt and respond to changes. So what they saw was that the rats in the loss group were unable to regulate their behaviors to loss. They would either show exaggerated or reduced responses. But once the researchers blocked the brain from producing more PNNs, the rats were able to better regulate their behaviors. Now, it probably seems silly to look at rat models and not humans, so how can we translate it to human populations? Well, given that the researchers found a marked difference when targeting the PNNs, or safety nets, this shows that it can be a promising target for humans. If we know that decreasing the number of PNNs in the brain can help to better adapt to loss, then this invites researchers to create clinical interventions where they can find ways to try to decrease these nets. However, every study comes with its limitations. As I mentioned before, the researchers looked specifically at male rats and not females, which may introduce bias. Another limitation is that they oversimplified loss. They only remove positive stimuli, which may not align with the complex nature of loss in humans. Plus, although these animal models can be very controlled, the reality is they are not human models, so we must proceed with caution when translating it and trying to generalize it to humans. Despite the aforementioned limitations, this is still groundbreaking research in the mental health field. Smail and colleagues provided invaluable insights regarding the neurobiological effects of psychological loss, insights that should be further examined in future studies. It is a great step in the right direction to alleviate the impacts of loss, which is crucial in order to preserve people's mental health and well-being.